We present here a novel biomarker testing technology for out-of-laboratory settings that has the potential to significantly increase the number of tests without increasing healthcare costs. Modern medicine relies on biomarker tests, and the proportion of clinical decisions influenced by biomarker data is continuously increasing. To achieve a 3.5-fold increase in annual testing per head, healthcare spending had to be doubled. Nevertheless, the achieved level of 2.5 tests per head is far from sufficient for evidence-based medicine. To reduce the costs, it is proposed to move from centralized to distributed testing, by conducting tests in near-patient settings, using inexpensive portable instruments and consumables, easily available to individual users. This can reduce the involvement of medical personnel and infrastructure, eliminate unnecessary logistics, and at the same time, speed up the tests. Such testing is especially useful during a pandemic or for patients receiving chemotherapy or dialysis at home. Distributed testing imposes strict requirements on biomarker analyzers. They must be portable, affordable, provide clinical accuracy and sensitivity, and be easy to use by untrained people. To meet these criteria, we developed a novel fluorescence-based biomarker analyzer that exhibits greater sensitivity and accuracy compared with instruments based on light scattering. The question arises, why is fluorescence better than light scattering? First, the scattered light caused by the formation of immune complexes has the same wavelength as the scattered light caused by bubbles or impurities. Therefore, in this method, we measure a relatively low assay signal against a background of strong sample scattering. Besides, the relationship between the scattering intensity and immunoprecipitation is a saturating function that limits the dynamic range. Further, scattering intensity depends on concentration, the intensity of incident light, and experimental geometry. Therefore, the method requires a reference sample, which does not always fully reflect the experimental conditions. In fluorescence measurements, the excitation and emission wavelengths are different, and it is possible to select the excitation and emission wavelengths such that both Rayleigh and Raman scattering do not affect the measurements. In addition, the emission background in the red and near-infrared ranges is low, which allows measurements of fluorescence in the picomolar concentration range. However, the most valuable property of fluorescence is the ability to use the molecular characteristics of the fluorophore, such as the fluorescence lifetime or rotational correlation time, as a readout parameter of the assay. Because these characteristics are independent of excitation intensity, sample concentration, or experimental geometry, their use allows immunoassay measurements to be carried out in a homogeneous format. For example, the rotational correlation time of a molecule, which gives the rate of molecular rotation in a solution, depends on the hydrodynamic volume of the molecule and viscosity of the solution. This parameter can be determined from fluorescence anisotropy measurements. Fluorescence anisotropy is defined as the difference between the intensity of the vertical and horizontal components, normalized to the total emission intensity. For many types of fluorophores, it varies from 0.4 in rigid solutions when molecules do not move, to almost zero if the viscosity is low and molecules rotate rapidly. This empirical equation gives the relationship of rotation correlation time of a labeled protein with its molecular weight. The weight of the rotating molecule changes when the ligand forms a complex with a biomarker. The greater the difference in molecular weight, the greater the change in the rotational correlation time and hence the anisotropy. The anisotropy of a sample containing a mixture of free ligand and ligand bound to a biomarker is described by a simple linear equation, which shows that changes can occur in the range R free to R bound. Despite the great potential of fluorescence anisotropy for immunochemical measurements, it has not yet gained widespread use in medical devices because the method requires the measurement of two fluorescence characteristics, the intensity of the vertically and horizontally polarized fluorescent components, which requires either two detectors or a complex optomechanical design. So far, such measurements are usually carried out in a laboratory spectrometer. With our proprietary technology that allows 2 or more fluorescence properties to be measured with a single detector, and without any moving parts, we have been able to develop a low-cost, portable biomarker analyzer that ensures laboratory accuracy and sensitivity. The instrument can work in 4 modes: fluorescence intensity, fluorescence anisotropy, fluorescence resonance energy transfer, and radiometric, in both endpoint and real-time formats. Due to the absence of moving elements, the analyzer is resistant to vibrations and can work for up to 5 days without recharging the onboard battery. Measurements can be conducted in a standard optical or disposable plastic cuvette. The small sample volume requires only a few picograms of test reagent, putting consumable costs in the range of several pounds per test. The simple mix and measure format makes it easy to use at home. The capability of this technology is illustrated by a standard curve of IgG antibody binding assay with the use of fluorescently labeled protein A. Another example is the standard curve of the C-reactive protein test. In both of these tests, we got a good dynamic range, which gives us the accuracy of laboratory instruments. This example illustrates the operation of the biomarker analyzer in real time, which allows for measuring the kinetics of binding and dissociation of proteins. Let's now compare how this instrument looks in comparison with other point-of-care biomarker analyzers. Well, compared with microfluidics, it has similar accuracy and sensitivity but is much less expensive. It also outperforms light scattering instruments in accuracy and sensitivity, while remaining less expensive. An important advantage of this biomarker analyzer is multimodality, which distinguishes it from other devices of this class. It can be configured to measure the concentration of biomarkers in the fluorescence anisotropy, or fluorescence resonance energy transfer modes. It can also quantify the concentration of ions in the radiometric mode. Besides, the real-time mode allows us to measure protein binding and dissociation kinetics, which is useful for research. In conclusion, we note that this inexpensive and easy-to-use instrument provides laboratory accuracy and sensitivity and can be used in any out-of-laboratory settings by untrained users. The introduction of this technology can greatly increase the number of tests without a significant increase in costs.